Good morning, everyone. And today in Spiritual Adventures, I'm going to talk about the science of vaccines. Similar to the previous talk on the science of sanitary precautions, this one I'm trying to keep it as general as possible, and I am hopeful that everyone can follow along and maybe gain something. This is a very important and timely topic. Okay, so before talk about the science, let's talk about a little bit of the history of this. And here are these two fine gentlemen. May have heard of them, and they are considered the people who first developed vaccines. Edward Jenner is the person who developed a vaccine for smallpox by observing that if those who were infected with similar disease but very mild called cowpox gave them immunity from smallpox. So he essentially figured out that if he could get that to everybody, they would be protected against smallpox, which turned out to be correct. Then, of course, Louis Pasteur gave us a lot more than just vaccines. He developed the rabies vaccine, but also he came up with, we all know, pasteurization, and most importantly, the germ theory of disease, which revolutionized how we view diseases. But also, you must remember that he thought viral diseases were also caused by germs, and it took another generation of scientists to actually understand what viruses were all about. So here are I've listed four gentlemen, and now we are getting into the modern biological studies, and biology is a complex process. So often you don't have one person standing out like Pasteur or Jenner, but many people making small advances to understand it. And it is these four gentlemen I pointed out who are generally considered to be the fathers of the field of virology, who, ending with Wendell Stanley, finally showed what a virus was. And I like this because all these four are from different countries, and that is an example of modern science, how it proceeds. It's not just one person, one country, but it is an international endeavor. So what did they finally conclude? This is the important thing. Viruses are not living germs, but complex biological particles. In other words, they are like, I don't know, some hair, nails. So they're not living, but they are very complex. And when I say they're not living, really what I mean is that they cannot reproduce themselves. So here is the picture of the coronavirus, and you may have seen this before, this pretty picture with all these spiky things on the surface, and that is why it's called the corona, because of those spiky things. And those are important, which we will come back to. Those pink things that are sticking out, those spikes, are very important in what we are going to talk about. But also, if you look at the red skin, that is the membrane of the virus that holds what is inside it, which is the genetic material. In this case, it's RNA. So what the virus does is that it comes and invades our cells and dumps that genetic material into it, and that genetic material is really then becomes part of the cell, and that our own cell starts reproducing the virus and eventually dying as a result of that process. So the virus itself is just nothing but a conduit for the genetic material, and that's why I said it's not living. It cannot reproduce itself. It is our own cells that do the reproduction for it. Okay, so how do we then deal with this? First, we got to understand how our body deals with it. And here is how our immune system responds to a virus. And this is important because ultimately this is what we have been relying on all this time to protect ourselves. There is no cure for any of the viruses. We just have to rely on our immune system to deal with them. So initially, when you have the first infection, it takes a while for the immune system to recognize and then begin to respond to it. And so you can see that in blue, that is the initial response. It takes time. And when that blue peak appears, that is when we have inflammation and coughs and all of that in the case of the coronavirus. And then eventually, hopefully, we will recover from it, and then everything calms down. So then you have the purple shows that if you have a secondary infection, no problem. Now the virus is very well equipped to deal with it, and it clears it out right away, and you won't even know it, because now the virus knows, the antibodies know to recognize the virus and attack it right away. And they retain it in memory, and they retain it in memory for many, many years, so that in many, many years later, if the same virus comes and attacks it, you will be protected from it. How does our immune system do this? And this is here a diagram showing that. So if you start from the top left, the pathogen, immune cell, which one of the immune cells, which we call the B cell, sometimes called the white blood cells, if it attacks the pathogen, encounters the pathogen, and look at the way the pathogen is drawn with those spikes on it. And that is important. And you saw that in the coronavirus, those pink spikes that we drew, that's the same thing that are shown here. So the B cell attacks it, engulfs it, and then it takes those spikes, and those spikes are important because they are going to be called antigens. What does that mean? Those antigens are how our immune system recognizes the virus. So the B cell then keeps the antigen, because then another cell called the T cell, which is the one that begins the recognition process with the antigen, and then generates the antibody response. So now antibodies are created that will recognize this pathogen, and the immune system will respond with more B cells that can come and attack it and get rid of it. So there are other processes, as you can see below. Then there is, this has to go into memory cells that retain the memory of this antigen, the response to this antigen, and then plasma cells that produce the antibodies. But the basic response is this, and this is what we are relying on still to fight viruses. We don't have drugs for viruses yet. And this is our antibody. The immune system is what does that job. Okay, so now the question becomes, how do we try to respond to this? So what we try to do is that we create ways to teach our immune system to recognize the pathogen even before they come and attack us. And that is what a vaccine does. A vaccine is a way to teach our immune system to have this response, so we don't have to wait for that time period when it has to build that recognition process with antibodies and everything. It already knows that and will respond right away. So how do we create vaccines? So here are the two standard ways that they are done. First is what we call the attenuated vaccine, where we are going and maybe changing some of the antigens on the surface of the vaccine, and certainly remove the virulence, some of the virulence from it. So some of the genetic material may be removed and some of the antigens may be removed. And now you have a vaccine, a virus, which is still, we are going to call live, in the sense it can still invade our cells and get them to reproduce it, but they are harmless. They reproduce at a very low level, and they are harmless, and they are very quickly. They can be recognized by our immune system and respond to it, and then after that, they will be fine. So whenever you get a vaccine like this, you might have a mild response to it, but generally the body can handle it very quickly, and then we have protection from that virus for all the time. So this attenuated virus vaccines are the most common viruses for all that we know. Measles, mumps, polio, all of those things are created this way. And these are the most common vaccines that we have. There is another type of vaccine which is called the inactive vaccine. Here, you take the complete virus and then you subject it to radiation, and that radiation destroys just the genetic material. The entire vaccine otherwise is intact, but the genetic material is destroyed due to radiation, and then you use that to create the vaccine. And again, the immune system will now respond because it responds to the antigens on the surface of the virus, and all of those are intact. This is how we create flu vaccines. Flu vaccines are created this way, and that is a standard procedure for those. All right, now here's a problem. It takes a long time to do this. Typically, for a new virus, minimum 18 months, could be longer. And as you can see now, we are in the middle of a pandemic, and it's a long time to wait for us to get a vaccine to protect the population. So that is the problem. What do we do when you are faced with a pandemic like this? How do we respond to it? We don't have drugs. We have to wait for our immune system to respond and protect us. And sometimes it doesn't work and people die. So this is where now we have to come up with a way to do that. But there is another problem that is coming up, which makes it even worse. And that's this: mutations. This is all the different types of flu viruses that have come and invaded us every year. And every year we have a different one coming. So which is why you need a flu shot every year. You can't, like for measles or mumps, you can't just have one vaccine and then be done with it. We need this every year because every year it's a different one that is coming through. The coronavirus is not like the flu virus in terms of doing that much of mutations, but it is also a mutated virus, because the first time this happened was SARS, which came out in 2002, 2003. At that time, we thought, okay, it was a one-shot thing. But next thing we know, we had another one come, which was called MERS. And now we have the novel coronavirus. So it is not a mutation every year like a flu virus, but we are talking every 5 to 10 years, a new one is coming through. So this is not going to be the end of it. 5 to 10 years down the line, there could be yet another coronavirus that is coming through, like SARS and MERS came before this. So it is even more important for us to come up with a way to develop some way to combat this. Otherwise, we are looking at worldwide pandemics every few years now. All right, so what do we do? And this is where we're going to have to understand a little bit of the biology of the virus and how it invades our body to try to come up with a solution for this. Well, one solution, of course, is to do this. You take the plasma from people who have recovered from the virus and purify it, because it will have some antibodies in it, and then you can give it to patients who are suffering from it. Of course, the problem is that this cannot be scaled up. This is, and it is not. We are not even sure it will be effective because the antibodies don't survive for long. So even you take the blood plasma, if you are lucky, some of the antibodies may survive. And if you give it to the patients before and remember that the immune system will recognize this as a foreign antibody and may not even allow it to survive. So it will be cleared out right away. So it is not clear this will help, but it is worth trying, and people are trying that, because if it works, that could, at least for critical patients, may be able to save a few of them. But that's not a long-term solution to the problem. Okay, so what how does the virus invade the body? Okay, so here it is, the virus. So this is the coronavirus. This is a diagram of the coronavirus. You can see that, and you remember all those spikes on the surface? They are actually designed to grab the cells to invade them. So here it is showing the coronavirus grabbing onto our cells, the cell walls, and then what they do is they do something called fusion. They fuse with our cells and then dump the genetic material. You can see in the middle the genetic material is there, and that is the genetic material that's dumped into the cell. So this is the first step in the invasion. So can we do anything about this? Well, there is some indication. Okay, now this is really not very clear this will work, but there are some drugs that might slow this, and particularly the drug chloroquine could interfere with this process and might help. It's not, this is not a solution to the problem, and it might just reduce it a little bit, but that's the best we can hope for. But there is some evidence that can do that. But here's the other problem with it. Our president talked this up, and next thing you know, this happens. So please don't recommend anybody to take this, because chloroquine is found in is a pool chemical, and so you can buy it commercially, and it can be deadly. And this is not the only. There are other people also been now found to have taken it and had to be hospitalized after taking overdosing on chloroquine. All right, so let's go to the next candidate, and this is I would say a more promising candidate. Okay, so earlier we talked about how this invades the cell. Now, once the invasion, the genetic material is dumped into the cell. Now, what the cell is going to do is that this genetic material instructs the cell to assemble all the different parts of the virus. So here is a diagram showing that the cell now producing all the parts that are required, and one of the important mechanisms for that is that the cell will start producing this protein called a polymerase. Polymerase, here you can see that labeled as a virus polymerase, and it's labeled as a virus polymerase here to distinguish it from polymerases that our own cells have. They are an important protein that is used by all cells. So now this virus polymerase is produced from the genetic material, and that virus polymerase begins the process of creating the rest of the materials needed for a new virus particle. Okay, so the problem here, one potential way to stop this process is to interfere with the way the virus polymerase works. But the danger is that if you can come up with something that interferes with that, it will also interfere with the polymerases in our body, and that could be deadly. So if you can find a drug that will interfere with the virus polymerase only, but leave our own polymerases unaffected, then we have a possibility of maybe reducing the virulence of this virus. And yes, there is one, and that is remdesivir, and that drug shows promise to interfere with the virus polymerase, but not our polymerases. And the NIH started a clinical trial for remdesivir in February. So that showed promise, but again, here is what happens when our president opens his mouth. So he touted that, and the next thing you know is the company Gilead Sciences, which makes it, and they got overwhelmed, and they had to stop the trial because it was just, everybody was demanding this right away, even though it is just in a clinical trial phase. And so they have stopped it, and I have no idea what's going to happen, what the progress on this is. And even if this is successful, remember, this can maybe reduce the virulence. It's not going to stop it for everybody. and it's not a cure. It's all it can do is reduce the virulence just a little bit by interfering with the way some of the virus particles invade our body. All right, so what else can we do? Well, just to complete that process here, once the cell assembles all the different parts of the virus, they have to be put together. Then that's the assembly part. The different components have been manufactured now. Now they have to be put together, and they are put together in a particular part of the cell that is called ER. That is it is within the cell, but as a section of the cell. ER. Those of you who are familiar with your high school biology may recognize the ER as what is called the endoplasmic reticulum. That's where the virus particles are assembled. So there they are assembled, and then once they are assembled in there, they leave the ER to go to outside the cell, and from outside the cell, they go out of the cell itself to go invade more cells or invade other people's cells. Once this assembly process begins, there's no way to stop it. People have tried to look into ways that, and there's just absolutely no way to stop it. So you have to stop this before it gets to this point, and that's the challenge. Okay, so what do we do then? Well, remdesivir is still one possibility, but really the best possibility is this one. If we are going to assemble viruses, the normal way to assemble the virus is what is shown there. You can see in that earlier diagram, I had the spiky virus particle, and then it is engulfed by the B cells from our immune system. And what it does is that it pulls out the antigens, those spikes. That is, the spikes are the ones that are important for our immune system to recognize this virus. So the question is, can we just use those spikes only to create a vaccine so that the system will respond, rather than having to assemble the entire virus particle, because that is the time-consuming part. And because once you're assembling a virus, that thing can be deadly. So you have to be really careful, and we have to run a lot of safety trials and everything before we can make sure that this works. So, but if you can just use the antigen, that's those spikes, that's a much simpler process, much faster, and we may be able to get this a working virus for this process, for this disease. Well, the thing is, it doesn't always work, and so far there's been only one really successful vaccine created this way, and these are called subunit vaccines. And the only successful subunit vaccine is what is being created for the hepatitis B disease. So can we do this? Well, the promise here is that this is how they were able to create a vaccine for Ebola. So that worked when they rushed the production and they tried this, and it seemed to work for Ebola. So the question was, can we do this for the coronavirus? So here is that pretty virus again with the pink spikes. And here is the spike protein in greater detail. Remember that spike protein is what allows the virus to grab onto our cell walls and enter it. And so that is how our antibody recognize this virus as something to be destroyed. So now our thing is that, can we get those spike proteins out of the virus and then make a vaccine out of it? Well, the company that was involved in creating the Ebola vaccine actually started work on this right away in January to see if we can create this, and they have made great progress. And in the middle of March, they began to safety test the virus in some patients. So this is right now the best shot that we have. If we can develop, if this works, we may have a virus much faster than the normal minimum 18 months that we were taking to develop a vaccine for this. So here is the news article that said they had begun testing on this in mid-March, and just on March 30th, Johnson & Johnson and Moderna, the company that is testing this, signed a deal to produce huge quantities of this vaccine. That is, if we can get it by beginning in September. So if you think about it, January to September, 9 months. So that is now half the time that is normally required for a vaccine to begin. So this is a great progress, and if we can make this work, we may be much better prepared to handle what's going to come also in the future, these kinds of pandemics that come about. So that's what we have for now. So short of soap and water or sanitizer liquids, the two best bets we have right now are, if the remdesivir trial shows promise, then maybe we can reduce some virulence. But this new subunit vaccine that is being developed, if that works really well, that would be a much better result for this process, and we may be able to contain this pandemic and future pandemics that might arise, which are mutations of this coronavirus that is certain to come and hit us in the future. Okay, I hope that was educational. Not very spiritual, but at least I'm hoping that this will give you an idea, and also try to understand when news articles come and misinformation comes, you know what is the correct news that is being reported and what is just hype. And certainly if you see anything about chloroquine, you know that is going to be hype, or any other drug that people may be advocating is hype. These two are the best ones we have for now, and hope you find this useful. I hope to see all of you at the service later.